In order to make Cadet Hoya happier with this class, I decided to go ahead and start changing some of the naming systems that I'm using to make sure that you guys know which orders come in first, second, third, essentially what order the videos coming in, so you're not just randomly clicking around trying to find the one that makes the most sense. While it was actually a good suggestion from Cadet Hoya, I still wanted to give him some crap. So why is it that we have a nervous system? What do we even care about our nervous system anyways? Essentially, the nervous system is going to be the integration center for everything that our body is going to do, whether it's consciously or subconsciously, whether it's something that we're intending to do or it's reacting to an external stimuli, such as a lion coming in through the door. You're not going to have enough time to sit there and think, I need to move my quads. I need to stand up. I need, you need to be able to rapidly respond to changes in the environment. And that's essentially what the nervous system is going to be able to do for us. It's going to give us the ability to walk, talk, breathe, but it's also going to be kind of the central organization for how and why and when we're making energy. It's kind of a cool system to have. We really have two major divisions of the nervous system, I guess you could call them. It's kind of how people like to call it, but they're integrated, and it's not necessarily hard to divide them into two separate, but they're all parts of the same whole. So you're going to have a central nervous system, and the central nervous system, as you can see over on the left hand side of your screen, is going to be divided largely into the brain and the spinal cord. So think that this is within the axial skeleton, right? If you're going through Kin's 4460 right now, or if you're in my 4460 class, we've talked about the axial skeleton, and that's essentially where the central nervous system is going to reside. The next center over is going to be the peripheral nervous system. And the peripheral nervous system subdivides twice, essentially. So we have an afferent division and we have an efferent division. What you need to know is that the afferent division is used to approach the nervous system. So it is cell signaling into the nervous system. So that's why you see that there's somatic sensory nerves, there's visceral sensory nerves, and there's special sensory nerves. So we're taking the signal that's coming from the environment, we're taking our sensory input, putting it all the way into our central nervous system, whether it's our brain or our spinal cord, and then we're going to kick it back out and exit the system via the efferent division. So that's why you see these motor divisions, the somatic motor division and the autonomic motor division. We're going to talk more in detail about each one of these divisions, but essentially the somatic nervous system or the somatic motor system is going to be what's used to control our motor or our skeletal muscle, Our autonomic nervous system is what's going to be used to control all of those involuntary muscles. So think all your smooth muscle within your stomach and your gut that's causing the contraction of the colon in order to pulse food all the way around. Think that this is the heart, this is cardiac cells, this is respiration, such as your diaphragm contraction. This is all the things that if you had to think about them on a conscious level, your brain would look like the dog from Up. You'd constantly be like, oh, right? So that's why we have these two divisions. One that is almost conscious level and one that is subconscious level. In order to facilitate the signal either into the brain or out of the brain, so either via afferent neurons because they're approaching the brain and approaching the central nervous system, hence afferent, approach, right? Or efferent motor neurons, they're going to be exiting exiting the central nervous system. They're going to have general structure that's going to look like what's shown on the left-hand side. A good way to kind of remember this is to think about it as a functional unit. Think of it almost like how you receive or you used to receive TV signal back in the day. You would put a satellite up on the roof of your house. That's going to be your dendrites. Your dendrites are going to be kind of these finger-like projections off of the cell body that are trying to pull in signal from other places. You're then going to have a cell body, which is where the nucleus is going to be contained. And then you're going to have the axon. And the axon is actually going to be what is conducting the signal, what's sending the signal all the way down to the end so that it can communicate with the next cell. So if you think about it, you're going to be up here on the top side of this diagram, you're going to be receiving signal here, integrating that signal, making decisions about that signal essentially, and then sending that signal all the way down the axon length into these branches of the axon. Okay? What's super cool about the brain and really the human body is we essentially run on electrical impulses. We run on electricity. That's why it's so bad to have changes in electrical input into the body, like sticking a bobby pin into an electrical outlet, right? Our body runs on electricity. It helps that we're pretty much all water, so we have efficient neuronal work. We have efficient neuronal communication. There's two real traits to neurons. So one, they're going to be irritable. So they're going to be like the angry person sitting in the... No. Irritability means that they have the ability to respond to a stimulus and then convert that into a signal or into an impulse. So they have the ability to receive input and then give that input out as an output. Does that make sense? They're also conductive so that they can actually transmit that electrical signal. If they were rubber, then we wouldn't be able to transmit the signal. They might be able to be irritable. They might be able to take and internalize a signal and then try to respond to it. But if they're not conductive at the same time, we then can't pass that signal on to the next level, right? It's this signal that's going to be propagated down the axon. And that's what I'm trying to show you over here on the left-hand side, is you're seeing all those flashing lights are going to be signals that are going to be taken and brought in, internalized, and then sent back out. And it's extremely rapid in how it's going to occur. That electricity and the change in electricity is going to be largely decided by this concept of resting membrane potential and threshold signals. So at rest, all cells, whether it's a skin cell or it's a neuronal cell, are going to be negatively charged on the inside of the cell. So that means the inside environment is going to be more negative than the outside environment. Or it's going to be what? Polarized, right? Think of it like a magnet. A cell is going to be polarized. On the outside of the cell, it's going to be more positive than on the inside of the cell. That's largely due to an unequal distribution of ions across the cell. By the end of this lecture, well, really by the end of this week, you should be able to describe in detail how and what is occurring in this graph. But what I want you to notice for this slide specifically is look to see at the voltage. We're actually using voltage meters, and we're actually seeing that exact text occurring here inside of the neuronal cell. We're seeing that at rest, it's about negative 70 millivolts. So it is actually negative inside of the cell. This ability to maintain a resting membrane potential that is negative is going to be based on our ability to transport in and out ions and the ability to move positive and negative charges around the cell. So it's going to be due to permeability of the cell as well as the concentration of the ions near the cell. Just like when we were talking about the electron transport chain and how there's these chemiosmotic gradients, and when you got a lot of crap on the outside and not a lot of crap on the inside, the lot of crap wants to get to the not a lot of crap, right? You have this movement from high to low, whether it's temperature from high to low, which we'll talk about actually later in this semester, whether it's hydrogen ions, whether it's electrons, whatever it is, you want to try to move from high to low. Gradients work because of that process. And just like when we were talking about the electron transport chain, these cell membranes are going to be punctuated with these strange looking transmembrane proteins that are going to actually regulate permeability and efflux, so exiting and influx of ions, in order to maintain this osmotic gradient. So when ions are free to move, it means that the gates are actually open, right? So imagine it like a closed door and an open door. When the door is open, you can walk through freely. When the door is closed, you're not going to be able to walk through. So as we dive a little bit deeper, we're going to start trying to figure out why these things exist, why we have a concentration gradient that's occurring. What I want you to see on the left-hand side of this diagram is that you actually have the concentration, which is what mm is going to be denoting. It's going to be in millimolar, so that's a concentration of these various positive ions, potassium K+, sodium Na+, and calcium Ca2+, are going to be a little bit different than they are on the outside, right? So potassium's 5 millimolar, 142 millimolar for sodium. Calcium's about 5 millimolar on the outside. You look on the inside, it's less than 1 micromolar, so it's super low. Essentially, just like that last slide, what we're going to do to control and manipulate these membrane potentials in the ionic flow is going to be to open and close channels. So if we open a channel for potassium, which direction do you think it's going to flow? Is it going to flow from outside to inside? Or is it going to flow from inside to outside? It's going to flow from inside to outside. And why is that? It's because the concentration of potassium inside the cell is significantly higher than the concentration outside of the cell. Does that make sense? Cool. Same thing with the sodium. So how are we going to control sodium? If we open up a pore that allows sodium to move through it, is it going to move from outside to inside or inside to outside? It's probably going to move from outside to inside because you've got a higher concentration on the outside compared to a lower concentration in the inside. If you just opened a pore that allowed it to transmit across or be transported across, it would flow from high to low in both of these. So if we open a sodium flood channel, what's going to happen? We're going to have this massive increase in positivity or positive ions inside of the cell. You should be able to agree with me because we go from 142 to 10. So if I take and I rip this open right here, and I just make a small little incision that only sodium can fit through, sodium's going to go through it. And thus, you have the change in polarity, or you have a depolarization. So how is it that we're actually going to maintain a resting membrane potential? Well, we have this really cool active transporter that's called the sodium-potassium pump. So at rest, you're going to actually have almost a complete closure of all of the sodium channels. So sodium's not going to be largely allowed to move into a cell, right? However, there is a couple of potassium channels that are going to be open. So if you follow along on this left-hand side and we follow along this diagram, you can see that you actually have a transporter that allows for 3 sodiums to get into it. You then donate a phosphate, or you provide energy into a system or energy into an equation. So you have an endergonic or an exergonic reaction. It's going to be an endergonic reaction because you're going to be fueling the reaction, right? You're then going to have a phosphorylation. So you're going to have a change in shape of this enzyme that's going to allow for sodium efflux from inside of the cell. So that means we have a leaking of a positive ion out of the inside of the cell, making the inside of the cell actually move more negative. At the same time, once you release those 3 sodiums, you're going to be able to allow 2 potassiums to bind to the inside of that protein, and then it will change shape and allow for 2 potassiums to come back into the cell. So the problem here, or what's interesting here, is that if you allowed this to continually happen all the time, what would happen? You would have a change in concentration gradient. You would have a change in positivity and negativity that's occurring. So we can't constantly allow it to happen. But one way that we can also maintain the gradients in some way, shape, or form, or the negative and the positive or the polar nature, is that the numbers don't match, right? We're pumping out 3 sodium and we're pumping in 2 potassium. So it's kind of a neat way that our body has designed a way to control the polarity of the system. If we understand that there's a polar nature of the system, if we understand that these axons have a polarity to them, and we understand that we can actually change that polarity, then that's how we're going to conduct an impulse down the length of an axon. That's how we're going to be able to take and internalize a signal through the dendrites, through the cell body, and then further communicate that down the line to the next neuron or to the muscle or whatever. So essentially, these changes in polarity, these changes in ions conduct the electricity, or create is a better term, is create the electricity. They create the electrical signal. So once the axon becomes depolarized enough, it's actually going to transmit an impulse, and there's going to be a rapid wave of depolarization that occurs. So that means that the inside of the cell is going to become rapidly more positive, and then the next cell is going to become rapidly more positive, and the next cell is going to become rapidly more positive. So it's going to continue to move along, and that's what this diagram, or what this GIF on the left-hand side is trying to show you, is that there is a rapid depolarization, or a rapid propagation in signal down the line of the axon, right? And if you look at this as it cycles back around, imagine that this is the point of depolarization. So you're getting a massive increase in signal, and then you're actually immediately repolarizing. So as soon as you have a depolarization, you start to repolarize. If the system stays depolarized, then you can't send another signal. You have to have a resting membrane state in order to send the next signal. You have to get back to negative inside the cell, or else we can't continually send signals. So if we know how to depolarize a cell, how the hell do we repolarize a cell? One, it happens immediately. As soon as there's depolarization, you start repolarizing almost instantaneously. Well, the repolarization is largely driven by this very acute and rapid change in permeability of the cell, or the axon, to potassium specifically. So if you look on the left-hand side, you're having an influx of sodium. You're having a massive increase of sodium inside of the axon. So you're actually having high levels of positive ions moving into the cell, right? So we're forcing it to go more positive. During repolarization, we're going to have an efflux of potassium out of the cell. So we're going to move a positive ion out of the cell. Thus, we're going to be making the cell body, or the inside of the cell, the cytoplasm, if you will, more negative by losing positives. And it's actually the depolarization, it's the act of depolarization that creates these almost like rifts, these changes in permeability instantaneously to potassium. Something that's really neat about axons and action potentials is that they function on this all or none principle. So once the nerve is activated, that sucker's going, right? Once you hit threshold, there's going to be a depolarization. As soon as you hit threshold, it's gone, right? Like, it is happening. It's the exact same thing as pulling a trigger. So as you can see on the left-hand side, someone's pulling the trigger. It's an aftermarket trigger on a Glock 19. And you can see that as soon as he is pulling that trigger past its breaking point, you're going to be sending a bullet. There's the speed at which you pull that trigger is not going to change the rate at which that bullet leaves the barrel, right? The only thing it's going to do, the speed at which you're pulling that trigger, is going to change how fast the next round fires off. So it's the same thing with these axons. As soon as that trigger breaks, it's sending a bullet. It's sent. As soon as the next trigger break happens, next signal is being sent. And in order to imagine it one step further, that trigger reset. So as you break the trigger and bring it back to its break point, break back. Imagine it like depolarization, repolarization, depolarization, repolarization. Does that make sense? Cool. So why are we depolarizing? Like Dr. Hyde, I get it, dude. Like, we are moving electricity down this cell that's super long and super skinny. That's awesome. What the heck does it matter to me in exercise physiology? I'm not an electrician. I'm not an electrical engineer. Essentially, the entire reason that we care about this is because we want to be able to send a message from our neurons all the way down to our effector organs. So we want to be able to elicit a response. And that all happens at the very, very terminal end of the axon. So where the axon meets either a muscle or another dendrite or something is where all the magic is really going to be happening. So if you move down this axon, you're going to actually see that you have a nerve impulse that's being propagated down this axon and into the synapse. So this area is called the synapse, right? You're going to have a host of neurotransmitters that are going to be held all the way down here at this kind of bulbous end, if you will. Once that nerve impulse happens, you're going to have a sending of these neurotransmitters down to the bottom end of that axon. They're going to fuse with the cell membrane and release all their neurotransmitters into this little synaptic cleft, or this gap between the end of the synapse and the start of the next, whether it's axon, or not axon, but the start of the next neuron or the muscle cell or whatever. When those neurotransmitters hit the next line, that's when the next level of magic happens. So you're either going to have something called an EPSP, or an excitatory postsynaptic potential, or you're going to have an IPSP, which is an inhibitory postsynaptic potential. I dare you to say that like 25 times fast, because I can't say it once fast. These are going to either send excitatory or inhibitory signals. So they're either going to induce a more positive change or a more negative change. So if we depolarize the next cell with excitatory postsynaptic potentials, EPSPs, what do you think is going to happen? We're going to continue to send that message, right? I keep pointing like way away, even though I know you guys can't see me. Or we're going to force the next cell to become more negative. We're going to polarize it even more. So we're going to reduce the likelihood of it sending the next signal, right? So that's EPSPs and IPSPs. They function on two processes: one, temporal, and two, spatial. So if you receive a lot of input rapidly that's saying go, no go, send it, don't send it, you're going to take and internalize all those messages and go with the one that's more. So if you get a ton of rapid signals saying send it, 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 rapidly, what's your decision going to be? Send it. Same thing with the neurons, right? If you have a bunch of people from all over the world telling you to send it, then you're going to do it. So it comes down to essentially how short of a time frame there is between release and binding versus how far these impulses are coming from. If it's getting it all across, then it's going to be spatial summation. If it's getting it in rapid sequence, then it's going to be a temporal summation. And this is just a really cool image of how this is working. So you can see that you're going to have, well, wait for it, and then a neural impulse. So you're going to have synaptic vesicles that are going to bind and then essentially fuse with the membrane, release all their stuff into the middle. It's going to bind and then send a signal. Release all of its stuff, bind, send a signal. Does that make sense? So on to the next slides.